it is, we have to hide behind the application, right? So even in developed country also, they are doing research on systematic behind or hide behind the application. Otherwise, people don't give fund. Another one we have Dr. Noorhidayah Mamat, senior lecturer at Institute of Biological Science, Faculty of Science, where University Malaya. Hidayah obtained her first degree in Bachelor of Science with Islamic Studies, specialized in ecology and biodiversity from University Malaya. Ini from Academy Islam ni, under the Public Service Department Malaysia Scholarship. She then continued her interest in science, biology at the same university under the National Science Fellowship, Ministry of Science, Technology and Innovation. Oh ni banyak hutang ni, kena bayar something kepada MyBiz program. So I need you to contribute more in that. And then she worked as a research assistant to expand her experience in that field. And then in 2018, she completed her PhD and started her career as a part-time lecturer, and then lecturer in 2020. So we have two young, energetic people today. So I would like to invite first Dr. Azrizal to present the presentation. I give you maximum 20 minutes. Then we discuss after both of you presented your presentation. Okay, Dr. Azrizal, you start first.
Okay, thank you, Prof. Dr. Ahmad Ismail. I also thank you to co-moderator, Dr. Amal, for inviting me to join this webinar series as a speaker. Okay, Assalamualaikum and good afternoon, everyone. Okay, let me share the presentation slides first. Okay, can you see?
Yes.
Okay, good. So today, I want to share some knowledge on the issues and challenges in butterfly taxonomy with special focus on the genus Eurema. So actually, this sharing is just on the common challenges that you may face in doing a taxonomic study, okay, particularly on insect identification, okay, that need to deal with morphology. Okay, in taxonomic study of insect, the most important thing is species identification that you have to put in priority before you can establish their taxonomic identity. So you have to know how to identify, how to diagnose and describe the morphology characteristic of the species first. So the real challenge in taxonomic study is actually begin with the identification process, especially when there are conditions that can interrupt the morphological structure of the insect. So in entomology, identification of the insects, I can say is quite challenging, okay, even with the use of a well-established classification key, but still challenging. Unless you are expert in the group of insects, in the particular group of insects. So to identify the insects, generally it is done by looking on the morphological structures and characteristic of the insect's body. So for example, we can identify the insects by looking on their body shape. Okay, as you can see in the picture, there are a variety of the insect type. So they might have a rounded body, elongated body, or slanted body. And also the structure like antenna. So maybe they have a long antenna, short antenna, hairy or segmented antenna. And then the leg structure, the body coloration and pattern. Okay, this is the major criteria used for identification of most insect species. Okay, so so you have to know the specific color, the unique pattern on the body is very important. And also the mouth type, okay, to identify the feeding type, like chewing, sucking, or piercing. Okay, how about the butterfly? Okay, butterfly, unlike beetle, bee, ants, or grasshopper, Okay, they are quite different, okay, because butterfly is a wing dominant insect, okay, meaning that the wing are the largest and the most obvious structure on the body of the butterfly. And because of that, identification of butterfly is mainly rely on the structure of the wings. So this include the structure like body, like the wing coloration, the wing marking, venation pattern, or some of the small structure like a number of dots, size of discoidal cell, and the number of spaces between the vein. Okay, so what actually give the amazing color to the butterfly's wing? Okay, the color in butterfly wings are produced by pigmentation. Okay, this is for ordinary color like brown or yellow, and from the structural coloration, what we call as iridescent. So in butterfly wing, they consist a lot of tiny scale, okay, as you can see in the figure. So when you enlarge the part of the wing under the high magnification power, so you can see the scale structure and how they arrange to create a thin layer surface. So each of the scale, okay, they can function to absorb and reflect light at the particular wavelength, which then give a specific color to the butterfly wing. Okay, that's how it's work to produce the color in the butterfly wing, that you see a very colorful, very amazing color. Okay, this scale, they are very fragile, okay, and easily detached from the butterfly wing if you do the rough handling. So when you are dealing with the butterfly sample, try to avoid to this rough handling, as you might lose their color or marking pattern that are very important in the use to in the identification process. Okay, so what actually cause challenges in butterfly taxonomy? So you may find it challenging when you are dealing with the group of butterflies that reported to have remarkable similar in morphology, or the group of butterfly that that shows the presence of the cryptid species complexes, or they have a polymorphism, okay, or we call as a phenotypic diversity or mimicry, or the group of butterfly that shows a phenotypic plasticity, okay, or termed as a seasonal polyphenism. So here, those conditions I actually have a direct effect on the changing the morphological appearance of the butterfly, which then cause the difficulties in determining the morphological character for identification of the species. Okay, in butterfly with remarkable similar in morphology, some of the important characters used to identify or separate the species may ambiguous, as they sometimes share or have overlap morphological characters. Okay, for example, identification of species Graphium beticus, okay, as shown in figure A, and Graphium chironidus, okay, figure B, okay, quite difficult because they are sharing the same Okay, continue. Okay, for cryptid species, example of the group of butterfly that reported to have a cryptid species is skipper butterflies of the genus Eurema. Okay, as you can see in the figure, okay, the three species, the three individuals here, they are actually three different species, but their morphology look exactly 100% same in terms of coloration, wing pattern, and wing marking. Okay, polymorphism in butterfly is where a single species can exhibit or appear with a different phenotype or morphologies. So example of polymorphism in butterfly can be seen in the female of Papilio dardanus, okay, that exhibits at least 14 varieties in their appearance. So as you can see in the figure, they look different in wing color and pattern, but all of them are actually belong to a single species. And phenotypic plasticity or seasonal polyphenism is a change in a butterfly color, pattern, or form in response to changes in the temperature, humidity, and rainfall level. So this condition primarily occur in butterflies that live in the seasonal environment. Okay, for example, species Eumaeus atala. Okay, this species is found in the east of Florida, USA. So the left picture that have a green brown color of the forewing, okay, is found during the winter season, and while the right one is found during the spring season. Okay, Next, we will look on the issues and controversies in the taxonomy of Eurema butterfly. Okay, so why I focus on this genus? Actually, because I just want to share the finding on of my previous work done on this butterfly group. So this group of butterfly is among the commonest where you can easily found them in almost everywhere. Okay, if you go to the recreational park, urban park, or around your house, they are easily being spotted. They are very common. This butterfly group, they are a good example that are presenting the remarkable similar in morphology. Okay, as you can see in the figure, they are all look similar, but actually they are 7 different species. So in Malaysia, there are 9 recorded species of Eurema. Okay, however, only 7 species that are very common and easy to find, while the other 2 species are considered rare. Okay, this is based on my previous study record. Okay, because that 2 species are very hard to find in the field. The 2 species are Eurema electoria and Eurema brigida. And due to their highly similar in morphology, the species identification is hard and often cause misidentification. And their relationship status among the member is difficult to determine based on morphology characters. Okay, here are to show some morphological characteristics of the wing used for identification of the Eurema species. So the member of this genus can be recognized by the presence of apical black marking on the forewing upper side, okay, shown in the figure A here. So the difference in black apical marking can be seen on their pattern toward the basal part here. So it can be have a broad pattern or narrow pattern, or the pattern is continuous along the basal line here. And on underside of the forewing, the species can be identified based on the pattern of brown patch. Okay, the label as 5 here. So the brown patch can be either totally absent, okay, no brown patch, or they appear or present as a small tiny patch like this or like this. And also can be large and dark brown patch, okay, like this. For the hindwing upper side, the figure C, the important characters used for identification of Eurema species is the black distal margin. Okay, black distal margin can exist either as a zigzag shape or continuous shape pattern like this. And for hindwing underside, okay, the morphological character used for identification is the presence of basal spot, okay, located in the space 7 of the intervein. Okay, even though this genus have a well-established classification key, okay, developed by Corbett and Penderville in 1992, okay, but the character used for species identification shows some overlap characters that cause difficulties in species identification. Okay, among the 9 recorded of course lead to the subsequent issues or problem in taxonomy, like their position in taxonomy and also their relationship status. Okay, this is some of the issues on the variation of morphology in Eurema Pieridae, okay, from my study in 2015. So I have done study on the distribution and variation of Eurema species in Peninsular Malaysia, and the finding revealed that Eurema Pieridae show a variation of wing morphology with correspond to the different elevation of the sampling site. So the pattern of black apical marking, okay, you can see in the figure A here. Okay, the pattern of black apical marking of Eurema Pieridae, their typical pattern is supposed to be large and bright at the basal part. Okay, but when collected in a high elevation, okay, at a location of Bukit Larut, Perak, that have the elevation about 1250 meter from the sea level. So we found that the pattern has been reduced and narrowed, okay, toward the basal part, okay, just like this. So this actually, this variation actually cause the Eurema Pieridae to look similar in morphology to its congener, okay, Eurema blanda. So of course, this provide issues on identification for the species that cause conflict in identification process. And the variation of the Eurema Pieridae is not only reported in Malaysia. Some studies from other countries, for example, Pakistan, okay, they report that Eurema Pieridae is having the variation in number of cell spot when rearing to the different condition of the environment. And because of the variation,
tak boleh jalan, Azrizal. Boleh kalau ada masa sekejap lagi kita anybody can ask question in the chat box so that we can answer later. This is not too specific. This is it's a general, actually, not too detailed about the specific subject. But anyway, you can highlight some important in your subject. So Azrizal, kita tangguh you sekejap. Kita sambung dengan Noorhidayah.
Boleh, no problem. Right.
Okay, I start present my slide.
You can set you punya internet kalau sempat kita datang balik. Noorhidayah, do 20 minutes ya.
Alright, thank you very much, Prof. So I'm sharing my slide right now.
A lot more people interested in entomology. All right, so now you can see your slide. Well, can you continue?
Yeah, sure. Next slide, please, Dr. Ahmad. Thank you very much. All right.
Okay.
All right. So basically, this is you know, my outline for my presentation, my sharing for today. I will start it with introduction, then follow with then end up with the Q&A sessions. Okay. Next, let's go to the introduction part. Okay, next slide. Okay, good. So I know basically most of you, I believe that most of you are already familiar with this, you know, with this odonates, dragonfly. But yeah, to give some overview about dragonfly. So dragonfly is an insect from order Odonata. Okay, so they comprise of 3 suborder. One is extinct already, the Anisoptera. Then we have another 2. The first one is dragonfly, suborder Anisoptera. In Malay, we call it as the pepatong. Well, the other suborder is Zygoptera. We call damselfly or jejarum. So basically, these 2 dragonflies and damselfly always be mistaken by their look. But actually, they have different characteristics and also structures. So despite of that, we normally use the term odonates. Okay, sometimes you might hear me say odonates. Okay, so that is the inclusive terms when we referring to both suborders. Okay, so the damselflies, as you can see from this figure, it have a smaller the right figure. As you can see, they have a smaller body build. Okay, compared to broader and larger body of the dragonflies. So in terms we fly uniform and sizes, and the hind wing also they have a very narrow shape. All right. So in contrast to dragonflies, in which they have a different sizes of wing, okay, which is the forewing and the hind wing, they have the broader wing. Okay, and generally the dragonflies or Anisoptera, they fly faster compared to the damselflies. Okay, so the wing itself is actually very important clue. Okay, normally we use the to identify species beside using their colorations, we also use the wing. Okay, because it an important clue of an insect identity. Okay, so when at rest, as you can see from these figures too, the damselfly has closed wing while the dragonflies, okay, they are open wings. All right, so next. Okay, so Odonata are well known but often poorly understood insects. Okay, so they are the insects that people always notify about their appearances. I believe that all of you also can easily see the dragonflies because of the adult. They have large and also colorful body color and also the acrobatic flyers. Okay, they are sufficiently noticeable that they have received numerous folk names. For example, we can see in here in North America, they're also known as the mosquito hawk, they're also known as the horse stinger, and also snake doctor. While in Malaysia itself, okay, we often heard about people called as the sibu-sibu or sesibu or tuyuk, okay, in Kelantanese, okay, I hope I pronounce it right. Tuyuk by Kelantanese and also Putu Miliung, and Capung by the Indonesians. Okay, so next. Next slide. Okay, so despite of they are being so well known as a group, okay, so they are also have general lack of knowledge. For example, in here, as you can see from here, sebelum tu, previous slide. Okay. Okay, so general lack of knowledge. Okay, for example, they have their phylogeny and also the classifications, okay, which have proved from the research that they have difficult to understand. Okay, they also have the lack of knowledge in terms of the impacts of the organizations. Okay, and also they have the lack of knowledge of functions and species compositions. Okay, they are also facing problematic in taxonomic issues. All right, like the uncertainties in Zygospora classifications, and so the last one, they also have the knowledge in study, especially regarding the larvae and also the exuviae of the dragonflies. Okay, next. So when we're talking about the diversity of odonats, right? So there are about for more or less 6,300 species plus species, okay, of dragonfly currently worldwide, okay, and which it will continue to grow when the new species are found. Okay, so in Peninsular Malaysia itself, we have the diversity of dragonflies is quite high, which is we have for about 250 plus species of the total in the world. While in Borneo, okay, as expected, the family compositions within these two regions, which is within the Peninsular Malaysia and also the Borneo, the endemicity of Borneo, they are higher compared to the Peninsula of Malaysia. Okay, but from the number of the species found in Peninsular Malaysia, about 23 species had been listed as the threatened or near threatened by the IUCN. Okay, however, the diversity of odonates of Malaysia can considered as poorly because the data that we have is normally only based solely on the scattered record and also descriptions. All right, so next. Then we go to the importance of odonates towards our ecosystem. Why is it so important that we need to study off of these dragonflies? Okay, in here I'm sharing some of the importance of study of dragonflies, odonates. All right, the first one we have ecology impact. So this ecology impact I divide into two, which is the dragonflies also. First is known as the bioindicator, and the second one is known as the biocontrol agents. Okay, so in fact, from the name stage, okay, the larva stage towards the adult stage, the dragonfly has a significant positive ecological impact. Okay, because researchers, they are look at dragonflies as ecological indicators. Okay, the presence of dragonflies, they can indicate the quality of the fresh water, the quality of the environment. All right, so they also have showed that the odonates, okay, are likely to have the implication of the conservations of the aquatic microinvertebrates. Okay, so the second one in the ecological impact is the biocontrol agents. Okay, for examples, they can eat the mosquito. The examples of odonates, for examples, we have Crocotermia species. We also have Pentella flaviensis species. Okay, because as you know that the adult of dragonflies, they eat the adult of mosquito, while the larvae of dragonflies, they eat the larvae of mosquito. So they can control the population of mosquito, and indirectly they can control the dengue species can be spread. All right. So the second important is that the economic importance. Okay, so since I'm talking about the dragonflies, they eat mosquito and also they eat other insects. All right, so they can help the environment because of they can allow they can allow humans to reduce use of the pesticide to kill the pest insects. Okay, so this dragonfly also can be used to help to diminish diseases spread. For example, they have the disease spread by the mosquito, by the host flies, okay, or the deer flies. All right. For example, diseases such as malaria, yellow fever, and also dengue as well. All right. So the third importance of dragonflies, okay, to the human, of course, they provide charming visual appeal. Okay, same like Lepidoptera, butterflies, okay? So they are provide charming visual appeal for ponds, for streams, and also other freshwater body because of their beautiness and also the colorful of their body. But they are also in some country, for example, in Japan and China, they use dragonfly as a traditional medicine. And also, the dragonfly also have been caught for food in Indonesia. Right. So the last but not least, the importance of dragonflies is they can be inspired in the biomimetic micro-air vehicle. Okay, so in recent decade, okay, so the design and development of biomimetic MAV, we call it as a micro-air vehicle, they have gained increased interest in the global scientific and also engineering communities. So this study, this new, new biomimetic study, so this has given greater motivation to the researcher to study and understand about the aerodynamics, okay, that involve the winged insect. And here, the dragonfly itself, they demonstrate a unique and very superior flight performance, okay, than most of the insect and even the birds, okay. So in here, the odonates, they can provide a very valuable reference to the aerodynamic design and control of dragonfly inspired biomimetic micro-air vehicles. All right, okay. So next slide. Okay, then we move to the words that related to odonates. All right. So now the odonates are receiving worldwide attention as objects of research. Okay. So we have several, I list down several studies that we had done in the odonates work. For example, we have biodiversity study, we have taxonomic study. So in scientific field, as you know, the taxonomy is very important for us to have the correct identifications of organisms, okay, before we can further with other research areas, all right. Then we also have the systematics and molecular studies, okay, which these include the modern techniques of DNA barcoding, the genomic studies that nowadays they are using NGS technique, and also the molecular phylogenetic analysis, right. Then we also have the behavior and biomaterial studies. Okay, so as you know, the odonates, okay, the dragonflies are a good models for assessment of ecological questions on animal behavior. Okay, in particular because of their diverse reproductive mode, okay, and mating strategies. As you can see in the riverbed, you can see they have a different mating strategies, okay, that include the territoriality and also that associate with the mating and oviposition sites, okay. So the changes in behavior, okay, are the first response of most odonate species to the environmental alterations. All right, so this is the sums of the studies that we had done in the dragonflies. Okay, next slide. Okay, so here I pick up some of examples, the published article of mine regarding the odonates research. Okay, so the first one is regarding the biomaterial. Just now I mentioned about the biomaterial study, right? So this is the biomaterial works that demonstrate the unique and superior flight performance. All right, that can be inspired the biomimetic study. So the title is, as you can see from here, the morphological and mechanical properties of flexible resilient joints on dragonfly wings. Okay, for in this study I use rhinoceros species as a samples. All right, so next. So this study actually was done in Riken, Japan in 2018, where we want to study about resilient. All right, so resilient is actually resilient can demonstrate the extraordinary extensibility and elasticity. Right, so in this study we use combined techniques. For example, we use the LSCM, the laser scanning confocal microscopy, and also we use the SEM, the scanning electron microscopy, which is these two is to illuminate the structure and study of functions of the wing flexibility of the dragonflies. All right, so we also use the nano indentations by using the atomic force microscopy, AFM, in order for us to measure the elasticity of the wing. All right. And lastly, not to forget, okay, we also perform a protein analysis, all right, of the wing samples in order for us to show what is the important protein in the wing that responsible for the elasticity and the wing flexibility. Okay, so next. So this is basically the result, the image that that we get from the SEM, okay, the initial scanning electron microscopy. As you can see from here, this is the different wing of different species. We actually in this species, in this study, I studied about 3 species: Rhinocephala fenestrella, Rhinocephala biforata, and Rhinocephala perforata. Right. So this is to confirm that they have the 2 main joint. As you can see from here, the vein, they have two main joint. For example, they have the mobile, the red circle color, and also the blue color is the immobile joint. All right. So next. So that one just now is using the SEM. Now we are using the LSCM. Okay. So we have 3 different species. You can go next to next. We have 3 different species. All right. So this is LSCM, the laser scanning confocal microscopy. So as you can see from here, the bottom figure, the UV band, it reveals the presence of the blue fluorescence materials in the vein of joint. So this one is known as the resilient. So if there is a blue color, the UV band, so it means that that particular area, they have the resilient on that. Okay, so resilient, as you can see from here, resilient was found mostly in the mobile joint. Okay, just now we are talking about the SEM image. So they are mostly can be found in the mobile joints, where the cross joints, they meet the longitudinal veins. Okay, whereas they have a strong patches, as you can see in C. Okay, the patches of resilient were found within the nodules of both either dorsal and ventral surface of the wings. All right. So next. So this is the summary of resilient spike. We also study about the spike. Okay. Spike mapping of the Rhinocephala group. Okay. So we also include another two other species from the same family as an outgroup. So basically, you can see these 3. This 3 shows the phylogenetic comparison resilient patches and spike in densophlies. All right. So the branches in the tree, the phylogenetic tree, is actually shows the relationships of the taxa based on molecular. Then in here we use the CO1 gene. Okay. Cytochrome oxidase C subunit 1 gene. All right. So next. Okay, so basically, okay, as you can see from this figure, your left-hand side image. Okay, so this figure shows the surface of wing, okay, wing image by SEM, okay, for the 3 respective species of Rhinocephala. So this image showing that the surface of the membrane and the surface of the mobile joint and also the surface of the immobile joint. All right. So as you can see from this figure, the SEM image, they reveal the morphologies on the surface of the wing. Okay, for example, it's formed like wave-like patterns. Okay, as you can see from the first figure. All right, and also it reveal the globular nanostructure on the wing. All right. Okay. So on the right hand side, okay, just now I mentioned about SEM, the images from the SEM. The right hand side figure is actually the image that portray still the membrane, still the mobile joint and immobile joint, okay, based on the 3 species observed but under the AFM, the atomic force microscopy. So actually the wing of these 3 species are very small, really small. This is just about 2.5 centimeter only. But when we're using the advanced microscope, and we manage to see the nanostructure that plays the important roles in the flight ability of the wing. All right. So next. So okay, so as you can see from here, so this is the figure, okay, that shows the typical force displacement curve, okay, of the 3 sections. Okay, still the membrane, the immobile, and also the mobile joint, okay, for the 3 respective species of Rhinocephala. All right, so it can compare the stiffness of the wing. All right, so it revealed that they have the descending order of stiffness, where as you can see from this figure that the immobile is stiffer than the mobile joint. The mobile joint is stiffer than the membrane of the wings of these three species. Right, so it can be concluded that within these three areas of the wing, the immobile joint had a high modulus of elasticity values, okay, compared to the mobile joint, and also the other area is membrane of the wing. All right, so next. So this is protein analysis, the last method that we use in this study, which is the protein analysis. So as you can see from here, the amino acid sequences, okay, the amino acid sequences, it's play the main role in defining the mechanical properties at the microscopic level. So in here, we managed to get 15 amino acids, okay, that were found in all the samples, okay, which is we have the, as you can see from this figure, we have the glycine, we have the glutamic acid, and also the alanine. Okay, so and this figure also can show the amount of each protein of the wing in each species of Rhinocephala. All right, so next.
Banyak lagi ke, Daya?
Okay, dah lah.
Good.
Okay, okay, tak jugalah sebenarnya. Okay, tak apalah, saya teruskan. Saya try to short it cut. Okay, so this is the conclusion, okay, of my first related works. Then we move to the next slide. So this
baru satu. Okay.
baru satu. Okay. Sekejap. 2 minit saya selesai. So my second, next slide please. Sorry, Dr. Ahmad, thank you very much. Okay, so that slide, this is my new baby. I want to stress this out. All right, so this is my new baby. I managed to publish last month, August 2021. So this is actually the study about the taxonomic study, which we tried to solve the taxonomic problem within the female. Just now I mentioned about the lack of the study, right? So this is tried to solve the problem within the female in the Rhinocephala species. All right, so next. Okay, so this is the experiment are about. So they have a confusion, they have a lack of, correct identifications, okay, of the sympatric species. Then in here we use also multi approaches in form of morphology. We also use the FSM, geometric morphometric analysis, and also the molecular technique in order for us to resolve the taxonomy uncertainties. All right, so next. Okay, so this is the classical taxonomy that we provide, that we come up with the key identifications for the females of these three species of Rhinocephala. All right, so next. Okay, so that one is the second method. So this is the third method is that, so we have we use the FSM, right, in order for us to study more about the ovipositor of the genital appendages of the female. All right, so we use the morphometric measurement first. Okay, then second. Next slide. Okay, so this is, we focus on the what is this, the appendages of the Rhinocephala. All right, so we can see the cilia are different between these three species. Okay, so next. So this also we call it as a tooth, okay, ataupun gigi lah, okay, for the odonates to insert their, their eggs while they are laying their eggs, okay, in the substrate. All right, so we try to differentiate the difference of this shedding tooth. All right, so next. And then this is the stylus. Okay, the stylus also is the important characteristics, okay, in female, okay. So they also have a different characteristic, different structure in stylus, all right. So next. Okay, then we use geometric morphometric. Okay, so from this we can reveal that the three species, they are clustered into their own cluster. All right, so this geomorphometric can be suggest that we can use this
Kenapa? Putus.
Doktor, kita orang tak boleh dengar suara doktor.
Boleh dengar? Saya guna different
Okay, boleh.
Boleh dengar? Okay. So we can use this method lah. Alright. So next. Okay, so this is the last method, the last approaches that we use to try when we're trying to solve the, the taxonomic problem, which is we use the phylogenetic tree. In here we use 2 genes, which is CO1 and also 16S rRNA genes. All right, so next. Okay, so this is basically the study that had been done, had been published about the odonate research. And the other study also, this is the behavior study that had been done by my ex-FYP about the behavior study. Okay, first about the reproductive behavior, the second one about the behavior ecology at the breeding site. So we have different study of odonates, all right, that we can study in details. Okay, so next. Okay, last slide. So for summary about my sharing for today, so I really hope that this sharing will offer new insight into odonate research with the utilizations of combined classic and as well as modern tools and methods. Okay, normally we use combined methods, okay, and tools. And I also hope that this sharing also can prompt more investigations on the potentially vast aspect in such study to promote greater interest on odonates. All right, thank you very much.
Okay, thank you, Noorhidayah. Thank you very much. There are many questions here. May I know between butterfly and moth? Rizal, nanti you jawab. What cause variation in Eurema wings and the Azrizal?
Okay, thank you. Okay,
no, wait, I want to read one question. So that Noorhidayah, how the dragonfly managed to manage as a biocontrol agent, and then apa lagi, dia ada buat koloni dragonfly dekat lab tak? It's not that, I don't know, maybe we can do it. Then any particular reason for using wrinkle Sepa for your experiment? Is there any other species that, I don't know if you can compare, but this will take a long time to read. What is the step-by-step criteria to nurture butterfly and dragonfly in Malaysian context? For example, native or endemic plants that can attract them. Then how dragonfly work as a bioindicator. So that's all. Try to answer and very short. Dr. Azrizal, try to answer, short answer.
Okay, thank you, Muhammad. Okay, so I am answering the question raised by Wan Zaki. Okay, so based on my finding, do you know what causes the variation? Okay, actually, I did not carry out the further study on why they have a variation. Yeah, I just focus on the population diversity study. So, but what can I share here? Okay, most of the reason they have a variation because they try to adapt with the environment, with the ambient temperature, or maybe become invulnerable to the predator. Okay, that's some of the reason.
Okay, Dr. Noorhidayah. Thank you.
So I'm trying to answer first one from author Prof. Chung. Any particular reason for using rhinocephala for your experiments? Okay, first of all, I'm trying to move from the dragonflies because nowadays they are not quite a numbers of dragonfly research. So I try to move to damselflies, in that which I trying to cater with rhinocephala. And why rhinocephala species itself is because of the approved study that saying that they have the problematic taxonomic problem within the females of the rhinocephala. So that is why I chose the rhinocephala species. All right, so I'm trying to answer about the ada koloni ke tak buat dragonfly. I'm so sorry Azrizal, we don't have kita tak buat lagi koloni dragonfly, maybe in the next time. All right, so how a dragonfly managed to as a biocontrol agent, had it applied anywhere? Okay, in Malaysia there are lack experiment doing this. I'm trying to get honestly I'm trying to get a grant, stating this, I want to apply this dragonfly as a biocontrol to mosquito, but I'm very sad because it had been rejected. Okay, in Malaysia because that is not really popular, but in other countries they had started to use dragonfly to control the populations of pest insect, especially the mosquito. All right, so last questions I think about dragonflies is about, okay, ideal. So this is my student, I think. Dr. Daya, how about dragonfly works as bioindicator by changing their color? Oh no, that is not by changing their color. That is by the behavior and also the presence or absence of that particular dragonflies in that particular habitats. All right, thank you. I'm trying to answer it as fast as possible.
All right, thank you very much. There are so many questions. It shows that the interest is there. So that's why, Dr. Daya, you can develop further the field, and we need more, as I mentioned. Malaysia is the top mega biodiversity in top 10 mega biodiversity in the world, and then we have a very good structure and infrastructure and the rules and regulation in Malaysia. We have to work together, fight for the conservation and protection so that we have time to study. And we need more people. I expected, last time I used to say many times in many places when we talk about a marine park, I just say we need 200 experts about marine park in order to talk about marine park. Same thing with insects alone, we need 100 to 200 to start because it's very urgent. Experts to work on, or interested people to work on insects. So it's a long way to go if you don't believe in it. This morning I have a lot of discussion on the EIA report on the further development of the economics, politician want to develop the area and after COVID development, many things. So they look different way, I look a different way. I say it's okay, we meet somewhere so that we need to conserve or protect our nature. You know, I just found one area where we fully developed in the coastal area. If you look at every state, there is an investment for ports and infrastructure, high-rise building and so on. So this is in the sea, so in the sea. So you will see that in the future, and how to protect that, how far we understand about, maybe in the next series, Dr. Zaleha is not here, we can work on the microscopic organism in the coastal area that a lot more we don't know. So this is the thing we need to move forward. I like young generation to follow through. I'm retired already. It's easy after retire, you can make more noise. And then as what you said just now, Hidayah, about the grant, I will say the reviewer, if you are here, reviewer for the grants, just listen to me. I said how far you understand about the paper that you are reviewing. So sometimes you don't understand. What we know is to cut the budget and we approve yes or no. For me, every proposal should give them a chance unless we know clearly that it is wrong. That's why I propose to the government, put the 30% for the top government priority, 30% for universities, and last sekali 10% for everybody, researcher or lecturer in the university should get the opportunity to develop their research. So I would like to thank all of you for the your participation in this meeting, and thank you, Dr. Azrizal, Dr. Noorhidayah.